In biology, I will be doing sexual reproduction in flowering plants. That is first chapter, sexual reproduction flowering plants. So don't miss any part of the video because this video is a very important video. So let's start the chapter without wasting any time. So basically, the first thing that when you start this chapter, let me just write down below sexual reproduction. What is sexual reproduction? Sexual reproduction, as you know, is a kind of reproduction where two parents are involved. This helps in creating genetic variability, helps an organism to survive better and improve because of genetic variability. You are getting characteristics of both your parents, mother and father, right? So mother has some good qualities, father has some good qualities, you are going to take both of them. That is the importance of sexual reproduction. And in flowering plants, the sex organs of the plant, basically the part of the plant that is responsible for this type of reproduction, is the flower. So the first thing we are going to discuss in this chapter is going to be the structure of the flower, what all the flower contains. So basically, when you talk about a flower, when I talk about a flower, flower has accessory holes, accessory parts and essential parts. When you talk about the accessory parts, these are nothing but calyx and corolla, because these are just protecting the flower. I will draw the diagram and show you, don't worry. Right now just remember the names. Essential parts are pistil and stamen. So pistil is the female part and stamen is the male part. We are going to discuss about them in detail, but these are the two things that you must remember when we talk about a flower. A flower just has these two types of parts, and we are going to discuss each of the part in detail. Okay, so let's start now. So first I'm going to start with the structure of the flower, and first we are going to discuss about the stamen, because the stamen is the one which is responsible for creating pollen grains. The flower produces pollen grains. Those pollen grains are the ones which are responsible for making the seed in the later stages of the flower. So let's start. So basically, if you have seen a flower, below the flower, there is a structure like this. This is called as the calyx. This is called as the calyx. The base of the flower, the green color part which you have seen in most of the flowers, this part is known as the calyx, and below this you have the flower attached. Okay, so this is the base of the flower, the green color part. This is known as the calyx. Now, above this, above this is the right above this is the pistil of the flower. So pistil of the flower is right above this. Pistil of the flower is right above this. I'm going to draw it with yellow color. The pistil of the flower is right above this. This is the pistil. Pistil of the flower. This is called as the female reproductive part. This is the female reproductive part. Inside this it contains the ovary and the ovules. So this is the pistil. And next, when you are going to discuss the next important part of the flower are these are the parts, these structures which you are able to see, these are known as the stamens. These are nothing but the male reproductive part. You can see these, I have drawn two oval kind of shapes here. So these are known as bilobed because they are bilobed. I will be explaining you that in detail later, but right now just understand this is the structure of the flower. And obviously there are the petals. Obviously there are the petals present in the flower. Obviously there are the petals present in the flower. This is how our flower structure looks like. So basically this part is the accessory hole. This is not very much essential. This is just protecting the flower. Here it will be covered like this. Okay, just to show inside I have drawn it like that. So this is known as the calyx of the flower. calyx. This is the calyx. And when you talk about these pink color, this is also accessory hole, the pink color petals. So petals are known as corolla. Petals are known as corolla. And next, when you talk about these this part, this is the male part, this is known as the stamen of the flower. And when you talk about this one, this complete female part, this is known as the pistil, pistil of the flower. So these are my essential words, essential words or essential part of the plant. So this is the structure of the flower. Once again, I'm just going to write it down because I want you to remember everything then and there. Okay, so that's why I'm explaining it multiple times. You must know this is called as the corolla. This part is known as the calyx, and these are non-essential or accessory words. And then when you talk about this entire part, this is known as the stamen, and this is known as the pistil. So this is the complete structure of the flower. Now what are the functions of each part? What each part is going to do? So first we are going to study about the male. Okay, first we are going to study about the male part. Let's draw the male part again. Okay, so the male part is known as the, I drew like this, two lobes on top of it, and below I drew a structure which, okay, so below I drew a structure which look like this, correct? So this is the male part, and this is the one which is called as the stamen, correct? This entire thing is known as a stamen. Now let's just represent all the parts of the stamen first. Okay, we will name the stamens all the different parts. So if you talk about the top part of the stamen, if you talk about this top portion of the stamen, that two bilobed part, that is known as, I'll just color this separately. So this bilobed part are known as, this bilobed part is known as anther. So who's bilobed? Anther. Anther is bilobed. And then when you talk about the below portion, when you talk about the part which is seen here, this is called as filament. The stalk where the anther is attached to, that is known as filament. And this is attached to the, this portion here, it is attached to thalamus or petal. It can be attached to the thalamus or it can be attached to petal. If it is attached to petal, thalamus or petal. Okay, so thalamus or petal, it is attached. So this is the part. Now the most important part, when you look here, the most important part is this anther. So we are going to draw the structure of this anther now. So anther, as I told you, is bilobed. So if you see here, anther structure looks like this. This is one lobe and this is another lobe. So there are like this two circles. This entire thing is one lobe. This part is known as one lobe, and this is, look here, this is called as one lobe and this is called as other lobe. But if you try to see it clearly, it has like this kind of curved. Okay, it appears as if there are four lobes, but they are not four lobes. These are two lobes. This is considered as one lobe and this is considered as the other lobe. Okay, so the top portion, I have just cut this anther. I have just cut this anther. So anther looks something like this. I have just cut the portion of the anther. Now inside this, if you see, there are microsporangia. Inside this, if you see, there are microsporangia. These four structures what I have drawn, these are known as microsporangia. These are known as microsporangia. this. Okay, I'm going to again zoom the structure to you. I'm going to draw this little more bigger, or let me just zoom here. Okay, let me just zoom it, this portion. If you zoom this portion, what you're going to observe here is there are different layers here. Okay, this is a sporogenous tissue. This tissue is this is a sporogenous tissue. This is a sporogenous tissue. This is a sporogenous tissue, and here there are multiple layers. Okay, so basically if you see the outermost layer, if you talk about the outermost layer, what you're able to see here, this outermost layer is called as epidermis. Epidermis. In entire biology you are going to see everywhere the outermost layer is generally called as the epidermis. Then you have the endothelium. Then you have this, then this portion. This portion is the endothelium. And next, inside you have a middle layer. I'm going to represent the middle layer with this orange color. This is my middle layer. Middle layer. And next, when you see inside, there is tapetum. Inside there is tapetum. This portion is known as tapetum. So these are different parts of the flower. If you are going to talk about the different parts of this flower, these are the different parts of the, not the flower, these are the different parts of the anther inside the microsporangia. Okay, this is microsporangia. This whole circle what I have drawn is microsporangia, and these are the different parts that I have labeled here, which you can see clearly. Next, now let's see how the pollen grain is produced. Okay, because that is what is very important. Inside the microsporangia, definitely the pollen grain is produced, as you know. But now, how is it produced? Let's talk about that. So there is a microspore mother cell. So there is a microspore mother cell. So the microspore mother cell is like this. It is diploid. It is 2n. The microspore mother cell is 2n. It's diploid. Then we have this microspore mother cell, basically it undergoes meiosis. The microspore mother cell undergoes meiosis, meiotic division basically. So this is meiosis. It is a reductional division. So meiosis it undergoes because it is diploid. And then you have studied in cell division chapter. Basically what happens, I will tell you. When 2n, that is diploid, multiplies, it gives rise to 4 haploid cells. After multiplication, see, in mitosis 2 are produced, but when you talk about meiosis, 4 cells are produced. So this one microspore mother cell, it is called a mother cell because it gives rise to other cells, right? So microspore mother cell, this microspore mother cell gives rise to microspore tetrad. It is called tetrad because 4. It produces microspore tetrad. Tetra means 4. So microspore tetrad, which are all n. Haploid, 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 haploid. It produces haploid cells. Next, these undergo again dehydration. Dehydration. Dehydration means, you know, whatever water is there, water from them comes out, and after they dry, they become what? After they dry, these 4 become our pollen grains. So they become little tiny, and they form, all are again haploid. All are again haploid. They form haploid pollen grains. So they are going to form haploid pollen grains. Next, what happens is, this pollen grain structure I am going to show you. This one single pollen grain structure I am going to show you by zooming it how it looks basically. Okay, so let's draw the structure of the basically the pollen grain looks something like the pollen grain looks something like this. It is going to be like this. Then there is a circular structure here. There's a kind of circular structure here, and just this part. So it looks like this. This is my pollen grain. I'm just drawing the parts of the pollen grain once. The outermost layer you could see the pollen grain. I have drawn it yellow right there. So here also I'm going to draw it yellow. Like this the pollen grain looks. It is having an outside a strong layer which is made up of sporopollen. This is nothing but exine. Outermost layer is called as exine. It is made up of sporopollen. That's why it is very strong. Sporopollen. Okay, so sporopollen. Next, inside if you are going to see, this is intine. The orange color portion what you are seeing, this is known as intine. And then you have vegetative cell and a generative cell. Inside this there are two parts. Basically there are two parts inside this. This smaller portion what you are able to see here, this is known as a generative cell. This is my generative cell. This is my generative cell. And the top portion what I have drawn, the top portion is known as the vegetative cell. Vegetative cell. And then exine you understood. This is intine. Intine is made up of cellulose and pectin. You must remember this. Exine is made up of sporopollen. Intine is made up of cellulose. Just like other parts of the plant. Cellulose plus pectin. It has pectin. So cellulose and pectin. Vegetative cell and generative cells be studied. And the diameter of this, generally if you see the diameter of the pollen grain, the diameter of the pollen grain is generally 25 to 50 micrometer. So they are very small, they are just like powder. But this, all this it contains inside it. Though it looks very tiny, these are the different parts it is going to contain inside it. I just forgot to draw the nucleus of this vegetative cell as well. So this is the nucleus of the vegetative cell. And this part is known as the germ. Why I have drawn a protrusion outside is because it helps to germ. Okay, so this is the entire structure of pollen grain. Now this basically this generative cell is the one which gives rise to gametes. So it undergoes mitosis and it produces gametes. Mitosis. It gives rise to male gametes. This symbol is for male. So it gives rise to male gametes. Male gametes or gametes, whatever you call it. So it gives rise to male gametes. And 60% of the angiosperm, two-celled stage. In 60% of the angiosperms, there is two-celled stage. In remaining, pollen shed at three-celled stage. So it is nutrient-rich and allergic. Pollen grains are allergic. All of you know that, right? For some people, pollen grains can be allergic. Now, pollen grains are male gametophyte. Their viability, basically, they are viable. Wheat and rice, they are 30 minutes viability. And for Rosaceae, Solanaceae, Leguminaceae, they can be viable for months. They can be viable for months. So this is about the pollen grains. So I will just mention the important point here. So their viability basically is pollen grains. Their viability in wheat and rice. Wheat and rice, they are 30 minutes. Just after the pollen grain is released, within 30 minutes they have to go and reach to the female part. And in Rosaceae, Rosaceae, Solanaceae families, and Leguminosae. For all these, it is months. They are viable for months. Okay, this what I have written is viability. How long they are viable. So this is about the male part. Okay, this is also known as microsporogenesis. Pollen grain is microspore, right? So this entire process, if I want to give the name to this process, let's give a name. This is known as microsporogenesis. Okay, so this is the part. I am going to make you to learn everything here only. You don't have to study anything later. I want you to master everything while I'm teaching it. So let's just give a short again. What all did I teach you? This is the part. This entire thing is the male reproductive part in the flower, and this is known as what? Stamen. And the top part of the stamen is called the anther. It is bilobed. It has two lobes. You can see this is one lobe, this is the other lobe, and when you cut it down, below it is attached by a filament, and down this entire stamen is attached below to the thalamus or the petal. Now when you see here, there are four microsporangias here. Okay, each sporangia I drew a structure and I showed you it consists of epidermis, then inside it is the endothecium, then middle layer, then inside it has the tapetum. They give rise to pollen grain. So there is a microspore mother cell which is diploid, that undergoes meiosis and gives rise to four microspore tetrad, which is known as microspore tetrad because it produces four haploid cells from a diploid cell, and those after dehydration form pollen grain. And the pollen grains are viable for 30 minutes in wheat and rice, and Rosaceae, Solanaceae and Leguminosae for months it is viable. If you see the structure of the pollen grain, the outermost strong tough layer is known as the exine, and inside it is having an intine with the germ pore on it. The diameter of the pollen grain is 25 to 50 micrometer. It has vegetative cell and a generative cells. Generative cells after mitosis, it produces gametes. So this is the entire process which is known as microsporogenesis. Now we are going to study about megasporogenesis. Mega. Okay, so the word itself, mega. So let's talk about megasporogenesis. Megasporogenesis. This is the production of female gamete inside the ovary. Ovules are produced, right? So the which color did I represent it by? I represented it by yellow, so I am going to draw it again by yellow. So there is no particular color which it is having. Generally these parts are white. So this So this is the stigma. The top portion what I have drawn is the stigma, and the style is the long portion, and this circular portion, spherical basically or oval shaped portion below what you're able to see, this is known as the ovule. Okay, stigma, style and ovule, and inside it is having ovary, and below it I'm calling the placenta right now, and ovule, ovary. So these are the parts. Okay, so basically if you see, the top portion is known as the stigma, where the pollen grains are going to land. Pollen grains fly and come like this and stick on the stigma, and this is the style. The pollen grains pass through the style. The pollen grains are going to pass inside through the style, and then finally they are going to meet inside the ovary. Okay, so ovule, ovary, placenta. So these are the parts. Now let's study about inside the ovule. If I'm going to study what is happening, okay, so let's study about it. So megasporogenesis is little complicated. I'm going to try to simplify it and teach you. Okay, so there is different parts. When I'm going to draw the parts of this, see below there is a structure like this. Above it basically above it, it is having a structure. This is the structure. This one, the middle portion I'm going to represent with orange color. Here also this is the orange color. Okay, so basically this part is known as the megaspore mother cell. This orange color portion is the megaspore mother cell. Just like how we had their microspore mother cell, here we have megaspore mother cell, because now you can understand from the word mother that this is going to multiply. So this is going to be 2n, that is diploid. And here we have the nucellus. This portion is the nucellus. And then you have integuments. These two portions, what you are going to see, these are known as integument. And then this is the hilum, and below you have the funicle, hilum, and it is attached to the funicle. So megaspore mother cell basically 2n, this undergoes meiosis. So it is 1, right? So it is going to first undergo meiosis, and it is going to produce a stage like this, 4 cells inside it. Like this it is going to produce, and then this is going to produce a cell like this. Each one of that, okay, are going to split like this, then inside 4, and then this is known as the embryo sac, 8. This is known as the embryo sac, 5. So that is the process. Okay, what is happening here? Meiosis is going to take place, 4 nucleus stage. Here then the cells are going to split, and then each cell nucleus are going to dividing. Here you can see the nucleus. This is the process, and then finally you have the embryo stage. Now what is going to happen in so many nucleus? You're able to see here so many nucleus, right? 8 nucleuses are present inside a single cell. So how was the structure of that? So this one, basically this cell, the embryo sac one, I'm just going to zoom that and show you, okay, right now. So let me just zoom that and show you. So when you see that, see the orange color one, I'm just going to draw that for you again in a much bigger. So this is the cell. Here above this we have, this portion is known as the antipodal end. Here you have the 3 nucleuses. Here we have the 3, and below we have again 3. This is micropyle, and this is this side of the cell is present near the micropyle. I showed you the structure, right? Micropyle. So here this is present near micropyle, and the top portion is present near the. So these are known as antipodal. Antipodal cells. Antipodal. 3 nucleus, what you are able to see here. And then we have synergids. These two are known as synergids, which are 2 in number. And then we have one egg. The middle one is the egg, which is 1 in number. And then polar nuclei are present. 2 nuclei are present in the middle. These are known as polar nuclei. They are present 2 in number, and this is the entire structure. Then there is a layer present like this, separating them inside. Okay, so there is a layer which separates them inside like this. Okay, so these are the different parts. So chelazal pole, the top portion is known as chelazal. pole, this is present above, and this is micropyle. Below it is present in micropyle. So here double fertilization takes place. What is going to happen? Basically, what is double fertilization? I will just explain you. So first the zygote is formed. After the pollen grains come and fuse here with the egg, right? After pollen grains come inside and fuse with the egg, first zygote is formed. Then second, pollen grains can combine and they can also fuse and form primary endosperm nucleus, which is also known as PEN, P-E-N, P-E-N, primary endosperm nucleus. So primary endosperm nucleus is formed. This primary endosperm nucleus, basically, if you are seeing here, its development. So its development is going to proceed. Its development precedes embryo development. It gives nutrition. Precedes before embryo development. This is produced. Okay, its development precedes embryo development. Before embryo is formed, this is formed, and it gives nutrition. It's nutritious. Example, you have seen coconut water or coconut. So coconut is an example of this. That is called as PEN, that is primary endosperm nucleus. And then here if you see what happens later, embryo is formed by a process known as embryogenesis. Later when the embryo is formed, embryogenesis, the zygote which was formed, the zygote forms globular embryo, it is a structure like this if you are going to see, and then heart-shaped embryo is formed. Heart-shaped embryo looks something like this. Heart-shaped embryo is formed, and then mature embryos. Finally mature embryos, complete embryo, mature embryo. So this is the process. So mature embryos formed finally. So this is the entire step if you are going to see about megasporogenesis. Okay, so this is the entire step. Now let's talk about what is pollination. I already explained you might have understood by now. Pollination is nothing but, I explained you the entire process right now. So what is pollination? Pollination is the transfer. Pollination is the transfer of pollen. Pollination is the transfer of pollen grains from anther, right? From anther it is produced, from anther to stigma of the flower. And the first step is known as, so there are different types of pollination, okay? Different types of pollination are autogamy. In the same flower if pollination happens, the anther produces the pollen grains which is landing on the same flower. Next there is also geitonogamy, different flower of same plant but different flower. And third one is xenogamy, X. See here, X means what? From one to other, completely different plant. Xenogamy means different plant, completely a different plant. So autogamy means same flower. In same flower only the pollen grain is getting transferred. Different flower of same plant. Whereas if you talk here, different plant, completely different plant. Okay, here if you see examples, some examples are given. Example here is viola and oxalis, and here also some xenogamy, some example is given, that is papaya. Okay, so this is what you must remember. This is pollination. This complete process, whatever I explained you until now, these all were the important steps in pollination. So hope all of you have understood clear. There are little small concepts left over after pollination what is formed. I'm just going to explain them quickly to you. But what all did we learn? We learned about the flower, we learned about the accessory and essential parts, then I explained you about this one which is stamen, then I explained you about the pistil. So we learned about the stamen, the different parts, anther, filament, and this portion is attached, and then we learned about the process of pollen grain formation. Now, pollen grain, see, look here, pollen grain comes from here and it falls here. It can be in the same plant, which is known as, I explained you, right, autogamy. So the pollen grain will come here, the pollen grain slowly go inside. A pollen tube is formed. When the pollen grain falls here, this plant detects it and forms a pollen tube. That pollen tube grows and it transfers the pollen grain here. A tube is formed. The tube will go and transfer the pollen grain here, and then the pollen grain is going to merge with this part of the plant. Okay, the pollen grain is going to merge here with the ovary, the ovules inside it. Okay, and the entire process of zygote formation takes place then like this. So all these steps take place, and for nutrition, primary endosperm is produced because of the other parts. Okay, so this is the entire process. And pollination, the transfer of pollen grains from anther, autogamy, zenogamy, and xenogamy. These are the different types. Examples for these are viola and oxalis, and xenogamy example is papaya. So now we are going to study about the agents of pollination. Who is going to cause that pollination, right? So pollination has to be caused by somebody. Who is going to go and transfer the pollen grains? So agents of pollination are who is the main agent who's going to cause that pollination. How are the pollen grains going to go reach to the female part? Because you learned they are going to different plant altogether sometimes, right? So these can be biotic means living, or they can be abiotic. When you talk about abiotic, it can be wind or it can be by water. When you talk about water, it can be on surface or it can be inside water, inside. So when you talk about biotic example is nothing but bee. Bee is the one which dominates this. Generally it happens because of bees. Honeybee dominates it. Okay, and water, wind, when you talk about, the pollen grains are generally light. Because of wind, whatever transfer happens, these pollen grains are light. Okay, so pollen grains are light. And exposed stamen, the stamen must be exposed, right, so that it will go and fly in the wind. Exposed stamen. And sticky pistil, and feather stigma. Stigma should be like a feather. So these are the different parts. Water, when you talk about on surface, when it happens, example for this is Vallisneria, and example for this is Zostera. Inside water of the plants where it happens. Now, how to prevent this? Because there is a problem, right? If the pollen grains are always coming and landing on the same plant, then there is an issue. Then there is no use of asexual reproduction, because asexual means we want genetic variability, but if it is happening in the same plant again and again, that's a problem. So how to prevent that? Outbreeding devices prevent it. Plants have certain features which prevent it. Those are known as outbreeding devices. Every plant, most of the plants, they protect themselves against this inbreeding. The plant, what is that? We learned, right, about autogamy. So plants want to protect themselves against autogamy. How they are going to protect themselves? Autogamy and geitonogamy also they don't want. They generally prefer xenogamy. So how to prevent it? So there are different types. The first step most of the plants follow is production of unisexual flowers. Some plants, they produce unisexual flowers, like papaya I told you, production of unisexual flowers. The flower does not have both. See, the flower does not necessarily have to have both stamen and pistil. It has only stamen or it has only pistil. Some flowers have only stamen, some flowers have only pistil. So they produce flowers like that. Okay, so to prevent such kind of inbreeding. And then if you talk about non-synchrony, both are not happening together, the production. Okay, so they are non-synchronous and position different. The position of the flowers can be different, otherwise position is completely different, so that they don't land up on the same flower, or incompatibility. Sometimes same flower, stamen, the anther, pollen grains, incompatibility. So sometimes the pollen grains are not compatible with the same flower. They cannot cause the production of seed when it is happening with the same flower. Now let's talk about the embryo. We have the dicot embryo and monocot embryo. Okay. So let's talk about the structure of embryo. So this is the entire process what all we learned until now. Let's talk next about the embryo. So embryo, when you see, the embryo has below a structure like this known as the root cap. Root cap, which is enabling the embryo to go inside the soil and grow. It is going to push inside the soil and grow. Above it is present the radicle, which is going to grow, right? Then above it, again more above if you go, there is hypocotyle. And then we have the plumule. This part is known as the plumule. And then finally we have two cotyledons. These are known as cotyledons. This is dicot structure basically. So these are the two cotyledons. Okay, so these are the different parts of dicot embryo. These are the parts of dicot embryo. When dicot embryo is formed, this is how it looks. And when you talk about monocot embryo, let's talk about the parts of that. So in monocot embryo, basically monocot embryo looks little different, so you might find it difficult to draw it. So this is how it looks inside it. So this topmost part is known as the scutellum, and this portion is known as coleophyte. This is the shoot apex, which helps to grow the plant. Shoot apex. And here we have epiblast. These are the parts. Here if you see the radicle, this radicle is the one which is growing below the soil, and it is causing the plant to grow basically. This is the radicle, and here also there is a root cap. This is known as root cap, which is enabling the plant to grow below the soil, the embryo to go below. And here we have the coleorhiza. These are the different parts of the monocot embryo. Okay, so hope it is clear until here. Now let's talk a little bit about the seed and a little bit about the fruit, because those are small topics. We will just finish it fast. So seed. Next we have our seed. So what is a seed? Fertilized ovule. After fertilization, the ovule becomes the seed. After fertilization, the ovule becomes seed. They have seed coats, on top of it to protect it. You have seen mango and all. They have seed coat. Seed coat can be albuminous. Albuminous is generally case of monocots. Monocots generally have albuminous seeds. Albumin is present, lot of albumin inside it, which gives nutrition. Okay, and non-albuminous. Non-albuminous example is dicots. Usually dicots. Okay, and example for these monocots and dicots, all of you already know. Wheat. Wheat is albuminous seed. Maize, castor, and here if you see pea, groundnut, and orchids. So these are example of non-albuminous seeds. Now sometimes perisperm, which is also perisperm, is nothing but persistent nucellus. Nucellus can be left over inside a seed. Perisperm, which is nothing but persistent nucellus. Persistent nucellus. Nucellus can be left in the seed. So that is known as, example for that is black pepper. Black pepper. Nucellus can be persistent inside the leaf, inside the seed basically. And dormancy record. When you talk about the dormancy record, how long a seed can stay without growing. You can keep a seed. You can keep a seed with you for how long, until it completely dies and cannot give rise. When you talk about Lipinus arcticus, that is one of the longest. Lipinus arcticus, we have record. And another biggest example is Phoenix dactylifera. You will be surprised to listen now. How long? How many years can you guess? Not 1000, not 500, not 6000. It's 10,000 years. Until 10,000 years, Lipinus arcticus seed can stay without growing. And after that also it will start growing if you plant it. Up to 10,000 years you can keep the seed. And this seed can be kept until 2,000 years. So this is a big record, right? They can stay dormant until this stage. So this is about the chapter. So next we are going to discuss about, so hope all of you understood this. Next we are going to discuss about the fruit. Small topic. Fruit. So fruit is nothing but fertilized ovary. This is nothing but fertilized ovule. When the ovule gets fertilized, it forms a seed, and when the ovary gets fertilized, after the ovary is fertilized, it forms a fruit. Ovary. After fertilization, ovary forms the fruit. Inside the fruit, there is seed, right? That is nothing but ovule. So, same way. Here, what you studied. Ovary is outside. See here. The ovary is the outermost part, right? Ovary is the outside part. This is ovary. And inside it, we have the ovule. This ovule forms the seed, the orange color part. So, outside part forms the fruit, and inside part forms the seed. So let's study about it a little bit. There are different types of fruit. One is true fruit. It can be a false fruit, and there are also parthenocarpic fruit. Without fertilization itself, they can form fruit sometimes. That is called parthenocarpic. Example for this parthenocarpic is banana. Example of false fruit is apple, because they are not formed from the ovary. Apple, strawberry, and then we have cashew. And true fruits are, example is mango. So that is a true fruit. So what is apomixis? Two more points. Apomixis, and what is polyembryony? Sometimes more than one embryo is produced. Formation of more than one embryo is known as, at a time if more than one embryo is produced, that is known as polyembryony. Apomixis is nothing but formation of seeds without fertilization. This is known as Apomixis. Example is Asteraceae and grasses. These are the examples. So this is the entire chapter. We have just covered everything in this chapter. Hope you understood everything. If you have any doubts left, please comment down below. I will try to reply to you. And do subscribe to the channel, because this is the only channel where your teacher is going to be replying to all of you and ensure that we cover all the chapters in just 14 days. So hope you subscribe to the channel and don't miss out any videos, and do comment below at every video after you watch how you felt the videos, so that I stay motivated and upload the further coming videos with more and more energy.